Okay, hi. Say hi to the camera for me, please.
Hi.
Okay. So I want you to imagine a world free of inherited diseases or viral infections. A world free of cancer, leukemia, okay, or HIV. You know, I lost someone to cancer when I was 12, my grandma. And that moment really marked my life. From then on, I started researching about cancer. In fact, I was a researcher for 2 years at Universidad Peruana Cayetano Heredia, and I have attended seminars given by the creator of CRISPR-Cas. Her name is Jennifer Doudna from UC Berkeley. So, what is CRISPR-Cas? CRISPR-Cas stands for clustered, regularly interspaced, short palindromic repeats. I know that name is a mouthful, but it actually comes from the structure you can see right here. As you can see, this is a cluster made up of repeats. Repeat 1, 2, 3. And in between the repeats, you have spacer sequences. Spacer 1, 2, so on and so forth. They are regularly spaced. Each spacer derives from the DNA or RNA of a viral species that infects the bacterial cell. When that happens, the bacteria can cut a segment of that DNA and add it to this chromosome. Then, this RNA is expressed into something we call a CRISPR RNA molecule. That CRISPR RNA molecule matches this original DNA sequence. So, eventually, the CRISPR RNA combines with an enzyme called Cas, which is just CRISPR-associated protein, Cas. That Cas enzyme is an endonuclease. It's like a molecular scissor that is able to cut down DNA. Okay, so when this guide RNA combines with the Cas enzyme, it forms a machine that is capable of cutting down DNA. That's why this functions as the immune system of bacteria. So, how does it work exactly? What is the technology? Well, this technology has 4 steps. In the first step, scientists need to design an RNA molecule that can target the specific sequence you want to cut. In the second step, the Cas molecule, which is a molecular scissor, binds to RNA. Then, in the third step, this machinery looks, hunting down the specific DNA target you want to cut. Once it is cut, in the last step, you have repair enzymes that fix that cut. Okay? So, now that you understand how this works, I want to analyze some of these major applications. So, first, as you can see on this table, one of the most important applications is cancer treatment. Right now, there are many ongoing research experiments going on in several labs. In China, there are so many experiments trying to attack cancer in many parts of the body, such as the liver, the kidneys, the nervous system, etc. How does this work? Well, the researchers in China, you have Xian Zhen, for example, who has developed an RNA molecule that matches specific genes involved in tumors. So, you know, CRISPR-Cas matches the sequences and cuts down the gene involved in the cancer development, knocking down those genes. So as you can see here, for instance, you have the target sites that are attacked by CRISPR-Cas, and then you have some of the editing strategies. Most of them are knockout genes. So what CRISPR does is knocks down the gene involved in cancer. Okay, and there is another really cool advance. You heard about HIV. HIV attacks T cells. Okay? So there is an experiment going on right now in which you can actually break down the HIV RNA. You can cut it down and inactivate its replication in T cells. Okay? So I know this is really exciting now, and I want to conclude with a very important thought. We're talking about our genomes here. Basically, we're altering our DNA, and at the same time, we're altering who we are. So the creator of this technology, Jennifer Doudna, warns us about the use of CRISPR-Cas because of its ethical implications. Imagine being able to engineer humans with specific features or characteristics, such as a higher IQ, maybe different eye colors, or maybe you want your son or daughter to be taller. Well, right now we don't have the knowledge to know exactly what genes are involved in those features. But once we know, then with CRISPR-Cas we would be able to make those changes and edit those genes. And of course, this has a lot of ethical implications. So we have to be very careful with the use of CRISPR-Cas. So you have any question?
Is it 100% effective?
Okay, that's a very good question. The thing is that CRISPR-Cas works, but it has some side effects. And right now scientists are working to make it better, more precise, to avoid any type of collateral damage. Any other question?
Is there any successful case?
Well, yes, definitely. Chinese scientists have actually applied CRISPR-Cas in some embryos, and they have been able to eliminate HIV from those particular embryos. Yes. Also, it has worked really well on animals, and right now we are trying it on humans. And I believe that within the next 5 to 10 years, we will be able to find a cure for most forms of cancer or viral diseases. Any other question? No? Okay. Well, thank you very much. Have a nice day.